So when these hydrogen ions pass through these channels, they essentially activate the mitochondrial membrane in which this process is taking place, or it stimulates it. And so that activates the next complex, which is complex 4, where you're going to have hydrogen ions passing through. Again, you have activation then of ATP synthase, which now you have hydrogen. These electrons are in the form of hydrogen ions, and they're getting ready to pass through this ATP synthase. But it's at this point here in the cycle where you have to have adenosine diphosphate being used to open up this ATP synthase. And once the ATP comes in, it's going to allow this hydrogen to pass through, and that's going to phosphorylate again the ADP molecule, converting it into adenosine triphosphate. So this process continues over and over and over, and it terminates with the generation of between 32 and 34 ATP molecules. And that's stemming from one molecule of glucose in the process of cell respiration. So remember, in cell respiration we have glycolysis, which is an anaerobic process, and then we have the Krebs cycle, which is used to generate our FADHs, FADH2 and NADH and NAD+ that are used in this system here. And then the last component is the electron transport chain, which we just went through and discussed. So all of those make up the process of aerobic respiration. Now, catabolism also has a process known as fermentation of glucose to help to develop ATP for a cell. It's not as efficient as cell respiration, because cell respiration uses the electron transport chain, and that generates about 34 ATP molecules. Total molecules is actually going to be 36 to 38. So scratch what I said. You get 34 ATP molecules from just the electron transport chain, but from cell respiration you end up getting between 36 and 38. But through the process of fermentation, which is anaerobic, there's no oxygen being used here. So the environment in which fermentation is going to take place is going to be anaerobic. Again, we start out with the anaerobic process of glycolysis, where you have the conversion of glucose molecule into pyruvic acid. And then what happens next is that you simply have a fermentation reaction that's going to produce 2 molecules of ATP. So let's take a look. I'm going to blow up this little diagram, so hopefully you'll be able to see it a lot better. But in this particular pathway, what we're looking at is the conversion of pyruvate into ethanol. Okay, and so when there is no oxygen being produced, you essentially have the pyruvate being converted into acetaldehyde, and then the acetaldehyde is going to take up a hydrogen from an NADH molecule, and it's going to produce ethanol. And in some instances, there will also be the formation of gas in the form of carbon dioxide, okay, as well as the generation of 1 to 2 molecules of ATP. So first process is glycolysis. The next process is conversion to acetaldehyde, and then you have the waste products being generated of ethanol and carbon dioxide. So this is essentially a very general breakdown of how the process of fermentation takes place when there is no oxygen present. So that's the fermentation of glucose. Now, in the process of catabolism, the breakdown of molecules is not going to happen unless we have oxidation reduction reactions. And oxidation reduction reactions are simply paired reactions where electrons move from one compound to another. So we primarily saw this with the electron transport chain, where you have electrons transporting from those NADH molecules to other carrier molecules. And like I said, you don't have to know the details of these elements, but just know that something like compound A, which has an electron, can move the electron to compound B. And so this is referred to as an oxidation reduction reaction. When a compound loses an electron, we call that the process of oxidation. when a compound gains an electron, we call that the process of reduction. So that's what we mean when we talk about oxidation reduction reactions. So oxidation will take place whenever an atom, an ion, or a molecule loses one or more electrons in a reaction, and then that particular compound is going to be referred to as being oxidized. When you have a compound that gains the electron, we refer to that molecule as being reduced or undergoing the process of reduction. And inside of a cell, an oxidation reaction is always going to be coupled with a reduction reaction, which is why we call these oxidation reduction reactions. And here's just a diagram showing you examples of how that would take place. So say you have like a carrier molecule like that FADH2. Well, the H2s are essentially our electrons. Those two hydrogen are going to be our electrons. So you have essentially the reducing compound A, which means it's reduced because it has gained electrons. And if these electrons are given to another compound, then that compound is going to be reduced. But the compound A is going to be referred to as oxidized because it has lost its electrons. So it goes from being a reduced compound to an oxidized compound. So that's just one example. For this oxidized compound here, which is our oxidized compound B, we're seeing that it is going to go in the opposite direction and it's going to acquire the electrons from the reducing compound A. And if it requires them, then now reduced compound B has become reduced or undergone the reduction. But it originally started out as an oxidized compound B. Okay, so hopefully you see that this is a pair-share reaction where you have to have oxidation and reduction to make it a redox reaction. And this is the type of reaction that takes place in the electron transport chain. So the transfer of electrons in oxidation reduction reactions occurs when you have one compound that becomes oxidized by losing an electron, and you have one compound that becomes reduced by gaining the electron that was lost from the first compound. And so we also call this compound A here, in another way that we define it is we refer to it as the electron donor. The electron donor is going to be the reducing agent because it is giving away its electron to another compound. Compound B is going to be the oxidizing agent because it is going to be the agent that was originally oxidized, but now it's being reduced because it's accepting the electron that's coming from compound A. So these reactions, as they're occurring in cells, are also referred to as dehydrogenation reactions, and that's basically stems from the idea that hydrogen ions are going to be removed from molecules in order for oxidation reduction reactions to take place. So that's the end of the discussion of the electron transport chain. Please email me if you have any questions regarding that material because it is very complicated, and that has to do with all the material in this particular unit. Now we're going to talk about anabolic reactions where energy is needed in order to create structures and generate molecules inside of a system or inside of a cell. So all of the energy that is put into place to build these particular units are going to stem from catabolic reactions. So catabolic reactions and analytic reactions, they're going to occur at the same time. So your cells are always going to be building and they're always going to be breaking apart molecules at the same time. With anabolism, we're looking at the concept of biosynthesis, where you have biomolecules being formed. So this can only take place by using the energy that was developed through the catabolic reactions. And when you think of catabolic reactions, think of the glycolysis process, think of the Krebs cycle, and think of the electron transport chain. Now, with biosynthesis, you can have energy that's going to stem from the processes of photosynthesis, if we're talking about plant cells, as well as chemosynthesis, which is where most eukaryotic cells get their chemicals from to enable the development of these biomolecules. In a photosynthetic reaction, energy is extracted in the form of light as it comes from some sort of light source, which would primarily be the sun. And then that light energy is converted into chemical bond energy in the form of adenosine triphosphate as well as carbohydrates. So the use of these reactions to generate biosynthesis is incredibly important, and it is so and is able to be so for a phototroph, because that energy is extracted from light and then converted into chemical bond energy. So here's a diagram showing you some anabolic reactions and how they'll essentially take place inside the cell. And this is very common to our own cells. Okay, so outside of a cell, you have things that need to get inside the cell. So these are going to be things that you extract from different food sources, such as sulfur, phosphorus, nitrogen, oxygen, hydrogen, magnesium, iron, potassium, calcium, sugars, amino acids, nucleic acids, fatty acids. We all eat things, and those things get broken down into these small components and then have to be transported inside of the cell. Once inside of the cell can start to generate monomers or polymers, and once these particular structures are developed in the forms of thymine, adenine, guanine, cytosine, and uracil, remember all of these, thymine, adenine, guanine, cytosine, and uracil, those make up nucleic acids, and that makes up our DNA as well as our RNA. So we have the synthesis of these monomers, and once they're synthesized, then we can have the synthesis of polymers, which are just simply many molecular structures joined together. Okay, we also have the development of fatty acids, glycerol, different polar groups, as well as amino acids. And then there's NAG and NAM, which this NAG and NAM is only essential to prokaryotic organisms such as bacteria. And we'll talk more about NAG and NAM once we start learning about the structure of a bacterial cell. But NAG and NAM chains formulate a structure known as peptidoglycan. So in an anabolic reaction, NAG and NAM is used to build a peptidoglycan structure, which is a chain-like fence that surrounds a bacterial cell. In the process of an anabolic reaction, proteins are built from the formations of peptide bonds that occur between them. Lipids are used to construct membranes that surround the cell. RNA are used to construct or build ribosomes that are used to undergo the process of translation inside the cell to build proteins. RNA is also used to build messenger RNA. And then DNA is assembled in order to create our chromosome structure. So there's a lot of building that takes place inside of the cell, but none of this is made possible without the help of ATP generated from catabolic reactions. Okay, so the key thing to take away about anabolic reactions is the fact that small components are used to build bigger components, which are used to build structures for the cell. So now we're going to move into our talk about bacterial genetics and kind of understands really what genetics is and how it fits into the grand scheme of things in this microbiology class. With genetics, we're talking about the study of heredity and the study of how genes are passed down from parent to offspring. An organism's genotype is all of the genes that exist inside of its chromosomes, and that's going to dictate the proteins, behavior, structures, everything that makes up that organism. When we talk about genotype, we just simply talk about the genes and we're talking about genetic code. When we talk about phenotype, we're talking about the actual physical traits that we are able to see when we look at a specific organism.